The idea of using microfluidic devices in chemistry to grow new teeth may sound futuristic, but it is increasingly grounded in real science. Teeth, unlike hair or nails, generally do not regenerate in humans once lost. Yet biology gives us hints that regeneration is possible. Some fish and reptiles can replace teeth throughout their lives. The challenge for humans is to design systems that can guide stem cells, growth factors, and mineralization in a precise, controllable way. This is where microfluidics enters the picture. Microfluidics, often called lab-on-a-chip technology, allows scientists to recreate miniature biological environments with exact control of fluids, gradients, and chemicals. Imagine a chip where nutrients, calcium, phosphate, and signaling molecules are delivered in the right sequence, mimicking the microenvironment of a developing tooth bud. By controlling these gradients at the micrometer scale, researchers can tell stem cells when to differentiate into ameloblasts, enamel-forming cells, or odontoblasts, dentin-forming cells. Doesn't this sound like designing a tiny, programmable biological factory on glass or polymer? The chemistry is just as crucial. Tooth growth depends on the deposition of hydroxyapatite crystals, the main mineral in enamel and dentin. In normal development, proteins such as amylogenin orchestrate the size, orientation, and hardness of these crystals. On a microfluidic chip, one could imagine using engineered proteins, peptides, or even nanoscale templates to guide crystal nucleation and layering. Already, experiments in biomimetic mineralization have shown that it is possible to grow enamel-like and dentin-like structures in vitro, although not yet at the full functional complexity of a tooth. In a particularly intriguing direction is on human growth. Instead of growing a tooth in a dish and implanting it, what if microfluidic patches could be applied inside the mouth directly over a gum site? These patches could slowly release signaling molecules, stem cell attractants, and mineralization agents in carefully tuned patterns, guiding tissues to regenerate in place. Think of it as a smart bandage for missing teeth, where microfluidics acts as the delivery system. Early prototypes of dental scaffolds already exist, but combining them with programmable fluidic control could make them far more precise. The paradox here is that teeth seem dead compared to constantly renewing tissues like skin, yet with the right chemical cues and delivery systems, the dormant regenerative potential may be unlocked. Microfluidics offers the fine control that bulk methods lack: spatial delivery of factors, dynamic gradients, and integration with imaging for feedback. It turns a crude, add chemicals, and hope approach into a programmable pathway toward tooth regeneration. If this succeeds, dentistry could shift from drills and implants to regenerative engineering. Imagine a visit where, instead of placing an artificial crown, your dentist applies a chip-based patch that slowly grows back enamel, or even an entire tooth. A lesser-known fact is that some labs are already exploring tooth organoids, miniature tooth-like structures grown from stem cells. Microfluidics could help scale these into usable therapies. Teeth do not naturally regrow in humans because enamel and dentin lack stem cell-driven renewal like hair or nails. Traditional dental treatments focus on repairing damage, not regenerating whole teeth. Tissue engineering aims to overcome this by combining cells, scaffolds, and growth factors. However, the difficulty lies in reproducing the hierarchical structure of enamel, dentin, and pulp, which require precise spatiotemporal control of mineralization and cellular differentiation. Without advanced tools, regrowing a natural tooth remains elusive. Dental implants replace missing teeth, but do not regenerate natural enamel or dentin. It is like replacing a broken clock with a new one rather than repairing and regrowing the gears from scratch. Microfluidic devices can precisely control fluids, nutrients, and chemical gradients at the microscale, making them ideal for mimicking the conditions needed for tooth tissue development. By using organ-on-a-chip approaches, researchers can simulate dental stem cell niches. The chip environment allows reproducible delivery of growth factors, signaling molecules, and minerals, while dynamic flow prevents cell starvation and replicates physiological stresses. These features make microfluidics a promising foundation for dental tissue engineering. A tooth-on-a-chip platform has been designed to model pulp-dentin interactions. It is like building a miniature greenhouse that nurtures fragile seeds with exact amounts of water, light, and nutrients. Dental pulp stem cells and DPSCs and induced pluripotent stem cells, IPSCs, are central to tooth regeneration. In microfluidic chips, chemical gradients can guide stem cells to differentiate into ameloblast-like and odontoblast-like cells, which are responsible for enamel and dentin formation. The microfluidic setting allows simultaneous testing of multiple signaling cocktails, accelerating optimization of protocols for targeted tissue development. Control at this scale enables reproducible and scalable cell-based therapies. Microfluidic gradients of BMP2 and FGF have been shown to drive stem cells toward odontogenic differentiation. It is like teaching apprentices different skills by exposing them to specific mentors in separate classrooms. The key to functional teeth lies in mineralization, depositing hydroxyapatite crystals in precise orientations. Microfluidic chips can maintain supersaturation conditions and deliver ions like K2+ and PO43 in a controlled manner. By regulating flow rates and concentrations, mineral deposition can be tuned to mimic enamel's organized prisms, and researchers can test additives that influence crystal size and shape. The chip thus becomes a laboratory for replicating natural biomineralization, bridging chemistry and biology for tooth fabrication. A microfluidic calcium phosphate delivery system has been used to grow enamel-like layers. It is like laying bricks with perfect alignment by controlling the supply of mortar at every step. Beyond chips, microfluidics can be integrated into biocompatible scaffolds implanted in the mouth. These scaffolds deliver growth factors and minerals in situ, guiding cells to regenerate tooth tissues inside the body. Localized delivery avoids systemic side effects, and implantable devices could replace root canals with living, regenerated pulp. By maintaining controlled microenvironments, regeneration could happen inside the patient rather than in vitro, marking a radical shift from prosthetic to biological dentistry. A hydrogel scaffold embedded with microtunnels has been used to sustain stem cell growth in dental defects. It is like installing irrigation pipes directly into dry soil to make a barren patch bloom again. Enamel is acellular and does not regenerate naturally, making it the hardest tissue to replace. Microfluidics may help by recreating the transient developmental environment where ameloblasts originally produce enamel. Chemical delivery, pH regulation, and ion gradients must be coordinated precisely, while nanostructured scaffolds can provide templates for crystal alignment. Combining microfluidics with bioactive peptides might restore enamel-like coatings. But full natural enamel regrowth remains one of the greatest challenges. Bioengineers have used amelogenin mimicking peptides to guide enamel crystal formation. It is like trying to rebuild a glass window from dissolved silica, requiring both structure and timing. Microfluidic platforms can incorporate real-time sensors to track mineralization and cell health. These sensors detect pH, ion concentration, and metabolic byproducts, and data feedback allows adaptive control of the regeneration process. This closed-loop system can correct imbalances before failure occurs, while integration with imaging provides high-resolution maps of tissue growth. Such monitoring brings precision to dental regenerative medicine. On-chip electrochemical sensors have been used to monitor calcium uptake during mineralization. It is like having a fitness tracker for teeth that reports every heartbeat of regeneration. The regenerative process itself resembles a form of chemical computation. Cells interpret gradients and signals as inputs and output organized tissues. Microfluidic devices can be programmed to present sequences of cues, essentially running biochemical algorithms. This computational view helps optimize complex, multi-step processes like tooth growth, turning regeneration into a designed program, rather than a random outcome. The chip thus acts as both incubator and computer. Sequential delivery of Wnt and BMP signals has been shown to control tooth tissue development on a chip. It is like running a recipe step by step on an automated kitchen robot, where the timing of ingredients defines the final dish. The long-term vision combines organ-on-chip models with implantable scaffolds for full in-body regeneration. In the lab, tooth-on-chip devices will accelerate discovery of optimal growth conditions, while in the clinic, patients may receive scaffold-embedded microfluidics that regenerate natural teeth over weeks or months. Coupled with AI for adaptive control, these systems may achieve near-natural regeneration efficiency. The fusion of microfluidics chemistry and stem cell biology could replace prosthetics with living tissues, marking a shift from repair to true regeneration. Researchers envision personalized microfluidic scaffolds seeded with a patient's own stem cells for tooth replacement. It is like planting a seed with its own irrigation and fertilizer system pre-installed, ensuring it grows into a perfect tree